Now, there are some salient features of the HGP. This is there in your books as well. And it's a huge list. It has about 9 pointers. So, first thing being the human genome contains 3164.7 million base pairs or 3 into 10 raised to 9 base pairs. So that is the first salient feature. The next one is that the average gene consists about 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Now, dystrophin is a gene which codes, it's basically coding for a protein in the cytoplasm. This is the largest known human gene. Which is the largest known human gene? It is dystrophin, and it has 2.4 million bases. So it's IIT-JEE, it has 2.4 million of these. Whereas on average, a gene will consist 3000 bases only. But dystrophin being the largest, it has 2.4 million. Now, the total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 1,40,000 genes. Almost all, that is 99.9% of the nucleotide bases are exactly the same in all people. So we see a lot of variation among people. And the first thought that comes to our mind is our DNA is different from their DNA and therefore we are different. It is just that 0.1% that is different in each of us. That is why we have different features. Whereas 99.9% is going to be the same in all people. Now, initially, when they started the project, they thought that the total number of genes in the human genome or the human DNA is going to be anywhere between 80,000 to 1,40,000 genes. But as they started sequencing and they realized that a lot of these genes are same in people, the number came down to 30,000 genes in the human genome. Now, the functions are unknown for over 50% of the discovered genes. So you have 30,000 genes, okay, that are found, of which 30,000 means about 15,000 genes. We still don't know what is the function of these genes. They are there, but are they expressing a protein? What are they helpful in? We still don't know. So about 50% of the functions of the discovered genes, we do not know. Then less than 2% of the genome codes for proteins. So out of the 30,000, that is the total number of genes, but out of the total genome, okay, less than 2% are coding for proteins which our body needs. Apart from that, we still do not know what are the other functions. The repeated sequence makes up very large portion of the human genome. So which means that human genome is very large. Right? It's about 3 into 10 raised to 9 base pairs. There are many repeated sequences in this particular human genome, and they make up a very large portion of it. The repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. So let us say there is one repeated sequence A, T, C, C, G. This particular sequence would be repeated about 100 to 1,000 times in the entire DNA. They are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. So these could be, maybe they do not code for anything, they do not express any protein or enzyme or anything of that sort. But maybe it will give you some information about who were your ancestors. Maybe through evolution, this sequence is conserved. By conserved, I mean your entire ancestors would have had this sequence. Okay, so it helps in finding out the structure, evolution, though they don't have a direct function. Now, chromosome 1 has the most number of genes. It has 2968 genes. Here I've already told you chromosome 1, the longest time. It was the last chromosome to be sequenced. That is because it has the most number of genes, 2968 genes. And Y has the fewest. Y is nothing but the sex chromosome, X and Y, of which Y has the fewest number of genes, that is just 231 genes. Then scientists have identified about 1.4 million locations where single base DNA differences, also called as SNPs or SNPs, single nucleotide polymorphism, pronounced as SNPs, occur in humans. This information promises to revolutionize the processes of finding chromosomal locations for disease associated sequences and tracing human history. Let us say you have this particular DNA. This is, let us say, in 1 minute. This is person number one. In person number two, you have the same sequence, ATAG. So, as you can see over here, there is just one single base difference. Instead of C, this person number two has A. Everything else is the same in this particular location. So this is nothing but a single base DNA difference. This is called as SNPs or SNP. And scientists have identified 1.4 million location in the entire DNA where this kind of a pattern is seen, wherein there's just one base difference. So here, please remember what is SNPs. This is single nucleotide polymorphism. So I just want to show you a small video here on the HGP. After this, we'll go to DNA fingerprinting. Okay. You can hear the audio.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA. And DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the ATGC sequence of 2 pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of errors, and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.